Good morning, everyone. Thanks for joining us today for our MESH virtual seminar series. My name is Melissa Rogers. I'm Associate Director of Business Development and Industry Relations at the USC MESH Academy, the Keck School of Medicine. For those who are not familiar with the MESH Academy, we are a dedicated team at the Keck School of Medicine at USC with the mission to empower the convergence of research across disciplines to address challenges in human health and disease. We take a holistic approach and bring deep functional expertise to help USC researchers connect and collaborate, both internally at USC and externally with the life science industry, to exchange ideas, increase collaborative research, entrepreneurial knowledge, and to build relationships to create long-lasting value. All right, so for today's seminar, we are going to have 3 speakers followed by our Q&A. So in order to ask a question, we ask that you please raise your hand. And when you do so, we'll be able to call on you, give you permission to speak, and you'll be able to speak directly with our presenters. If you don't feel comfortable, you can still use the Q&A function, and I can read your question out loud. If you do that, please specify which speaker you are asking your question to. But we really do encourage you to raise your hand so that the seminar is more interactive. In addition, this webinar is being recorded and will be available on the MESH website in a few days. So for today's seminar, we are focused on organs on a chip, and we have 3 great speakers who are going to be presenting on the topic. So it is my honor to introduce our speakers today. Our first speaker is Dr. Megan McCain. She is Assistant Professor at the Department of Biomedical Engineering in Stem Cell Biology and Regenerative Medicine. And today her talk is entitled Engineering Microscale Disease Models for Heart and Skeletal Muscle. So please welcome Dr. McCain.
Okay, great. Thank you for the introduction. Let me go ahead and click over here. Okay. All right, so first of all, I wanted to thank the MESH Academy for organizing this event and inviting me to speak. It's really a pleasure for me to be able to share our work today in this format and also present alongside my colleagues, Shannon and Keyue. So I first wanted to acknowledge the students and postdocs in my group who worked hard to collect all the data that I'm going to show you today, as well as our funding sources who make the work possible. All right, so what you can see here in this graphic is an overview of the drug development process. So I first want to draw your attention to this funnel, which roughly represents the number of compounds that make it through each stage of the process. So usually this starts with hundreds to thousands of compounds that are first tested in a variety of preclinical models, which I'll elaborate on in the next slide. These are then narrowed down to maybe tens of compounds that would then enter clinical trials and be administered to patients. Hopefully at least one compound would then make it through clinical trials and then be successfully be able to enter the market. So what I want to highlight next is what some of these numbers showed. So this top row here shows you that both the preclinical and clinical stage of drug development each costs about $1 billion and take about 5 to 8 years. Another important point here is this third row that shows rates of attrition, which are relatively high at every step of this process. So what that means is a lot of compounds will make it through preclinical testing and seem like they're going to be promising and then enter clinical trials and fail. So this can be a huge loss for drug companies in terms of time and money and resources. And so it's really problematic when the further along compounds get in this process and then will fail. So why does this happen? So one of the reasons that for this kind of failure within clinical trials is due to the limitations of our existing models that we use for the preclinical stages of drug development. So early stages of drug development rely primarily on in vitro models. So these are generally cells, usually human cells, that are cultured in a multi-well plate. So the ability to use human cells is really an advantage of these in vitro approaches, but usually these are cell lines. So these are cells that have been expanded for many generations outside of the body and have really lost a lot of their native-like behavior. They also represent only one human patient, right? So they're not reflective of the diversity that we have in the human population. These cells are cultured generally on a plastic surface that's uniformly coated with matrix protein, which is very different than the environment that these cells experience in our body. So in all of our tissues, cells have distinct structures, distinct cell-cell and cell matrix interactions that are really critical for their function. They can also be exposed to different physical forces depending on what tissue they're in, so things like flow or stretch. So cells in vitro in these conditions really act very differently than they do in our body. So that can affect their phenotype as well as responses to drugs. So the advantages here of the in vitro models beyond using human cells is that we can have access to cellular phenotypes really in real time. So we can image them, we can do a variety of biochemical assays and get a pretty clear understanding of what's happening on the molecular level. They're also generally high throughput and low cost, so great for screening large numbers of compounds. Later stages of drug development will then use primarily animal models. The biggest limitation of these, of course, is that they're non-human. So there's just fundamental genotypic and phenotypic differences between animal models and humans. The advantage is also clear in terms of the entire organism is intact. So if we want to look not only at what's happening in cells and organs, but we can also look at things like organ-organ interactions and immune responses that we will likely never be able to fully recapitulate in vitro. Another limitation of animal models is, unlike in vitro models, we have really limited access to cell and tissue phenotypes in real time. So we're usually limited to kind of collecting snapshots of what's happening in the organism during different disease processes or drug responses. Animal models are also low throughput, high cost, and raise a lot of ethical concerns related to in vitro models. So together, what this really highlights is we need new preclinical models that can provide access to dynamic functional phenotypes, which is really the advantage of in vitro models, while also recapitulating key aspects of cell tissue architecture, which is the key advantage of in vivo models. And we also need preclinical models that are really reflective of human relevant genotypes, which neither model system really does a great job of achieving. So organs on chips are a new technology that has been developed to address some of these concerns. It's really exploded over the last decade. So organs on chips in general are surfaces or devices that have been engineered using a variety of biomaterials and microfabrication approaches that are designed to provide a microenvironment for the cells in vitro that mimic key features of their native in vivo environment. So the goal is by culturing the cells in these organ on chip platforms, they'll respond in a more physiologically relevant way. So they'll have more disease relevant phenotypes and drug responses that are more predictive of what we would expect in a human patient. Because they're still in vitro, we still get a lot of the advantages of an in vitro model in terms of we still have access to phenotypes in real time, right? We can use these for live imaging and really see what's happening on the cell and tissue level. And where organs on chips become really powerful is when they are implemented with patient-derived cells. So these could be cells extracted directly from a tissue, or what we use primarily in my lab are induced pluripotent stem cells. So these are stem cells that start out as somatic cells isolated from a patient, usually a skin fibroblast, which can be very easily isolated from a skin biopsy. These fibroblasts are then reprogrammed to become pluripotent stem cells or iPS cells. And these pluripotent stem cells can then be differentiated into a variety of downstream cell types, including cardiomyocytes, which I'll talk more about. With patient-derived cells, not only are they human relevant, but they're also patient-specific. So iPS cells, like I mentioned, we can generate just from a simple skin biopsy. So it's relatively non-invasive, so it's very easy to collect these from a wide variety of patients. Together, organs on chips and patient-derived cells kind of gives you moderate throughput, moderate cost, so something kind of in between conventional in vitro and in vivo models. And with these, you still can't get those organism level responses, right? To look at things like organ-organ interactions or immune responses is still very challenging with these models, but that's not really what they're designed to do. So in general, these are not meant to replace existing model systems, but more to complement them. So together, these technologies have become really a new paradigm for human relevant disease modeling and preclinical drug testing. So in my lab, we work primarily on developing and implementing models, organ on chip models of the heart. So for all organs on chips, really the first step is identifying what is the minimum functioning unit. So the main function of the heart, as we all know, is to pump blood. So the minimum functioning unit here is a piece of cardiac tissue or myocardium. So myocardium consists primarily of cardiac myocytes, which are the muscle cells themselves. They're elongated, they're aligned with each other, and they're embedded in a compliant extracellular matrix. So as these cells contract, they're all working together, and they're able to actually shorten and be able to pump blood from the ventricle. So these are really our design parameters. So when we're trying to engineer these organs on chips, then we want to mimic some of these features. So one technique we use a lot in the lab is microcontact printing. With microcontact printing, we're essentially doing rubber stamping on the microscale. So we first make silicone wafer templates in clean room facilities with features down to about 1 micrometer. So here you can see one example. This is a stamp made from PDMS, which is a silicone kind of rubber-like material. The square here is about 200 microns by 200 microns and has kind of internally aligned features as well. So when we coat these rubber stamps now with matrix protein and invert them onto our substrate and then seed our cells, in this case cardiomyocytes, they conform to the patterned features and form an aligned piece of myocardium that has similar structure to native myocardium. Another feature here is we can culture these cells on polyacrylamide hydrogels, which have mechanical properties very similar to the native extracellular matrix. So we're also recapitulating that key kind of functional feature of the heart. So the main function of the heart, as I mentioned, is to contract. And so we want to be able to actually measure the amount of force generated by these tissues. So to do that, we put fluorescent beads within the hydrogel, which we can then image as the tissue contracts and be able to quantify actual force generated by the tissue. So instead of relying on kind of indirect metrics like gene or protein expression or just beat rate, we have a really nice physiologically relevant quantitative metric from these tissues. We've been adapting these now to iPS-derived cardiomyocytes to make human micromyocardium. And here you can see an array of these tissues with a calcium indicator dye that is fluorescing every time they contract. So you can also see in this movie kind of the reproducibility that we get when using microcontact printing. So every tissue has more or less the same architecture, and we're able to make a lot of measurements kind of in parallel by using these micro patterning approaches, which is another advantage. So my lab also works on skeletal muscle. So skeletal muscle are the voluntary muscles in your arms and legs. These are activated by motor neurons that branch off of the spinal cord and form synapses with the muscle fibers. So there are many different types of neuromuscular diseases where there's some dysfunction in one or more components of this unit, such as ALS. And these diseases in general are incurable. And one reason, again, is the challenge in generating preclinical models. So these diseases have a lot of genetic diversity. So for ALS, for example, these are genes that have been known to cause the disease. And even within each gene, there's multiple mutations that can change the type of disease or severity of the disease. So it's very difficult or kind of impractical to generate an animal model that captures every single type of mutation that we see in patients. So there's been a lot of progress in the field in making motor neurons from iPS cells that begin with patient-derived fibroblasts. But to really study dysfunction at the neuromuscular junction specifically, we need to be able to culture these motor neurons with muscle tissue. So the problem is in vitro muscle tissues have kind of lacked the structural and functional maturity that you need to be able to form mature synaptic structures. So my lab has advanced the use of these micromolded gelatin hydrogels for engineering muscle tissue. So this starts with a droplet of gelatin, which is derived from collagen. We can make a hydrogel, mold ridges into the surface, and also use a laser engraver to cut cantilevers into the hydrogel. So when we culture muscle cells onto these cantilevers, we can then peel them up and actually track the deflection of the cantilever as the muscle cells contract. So here you can see how we use this platform to compare 3 different muscle cell sources and compare their structure as well as their function. So these are now these cantilevers contracting in response to electrical pacing. So the chick muscle here is what was strongest. And so then we proceeded to co-culture those with iPS-derived motor neurons. And you can see, especially at the 3-week time point, the green here shows you motor neuron, and the white here is a synaptic marker on the muscle fiber. So as the motor neurons and muscle fibers start to strengthen their connection, you expect these structures to become more co-localized and more kind of invaginated with each other, which is what we're seeing after 3 weeks and closely approaching what we see in mice. I should mention also the iPS-derived motor neurons were provided by our collaborator, Justin Ishida. We also worked with Dion Dickman to measure electrical responses within the muscle fibers that are being driven by the motor neurons as another kind of functional output here to look at maturity of the neuromuscular junction from a functional perspective. So putting this together, we're seeing kind of structural and functional maturity of these tissues that we haven't seen in kind of previous, more basic in vitro models. Okay, so this is my last slide. I'm happy to talk about this offline or during the Q&A session, just some other work going on in my lab. So we've also been layering our hard-on-a-chip assays on top of a microfluidic device that can generate oxygen gradients to mimic the local deprivation of oxygen that you see in a myocardial infarction. We're also adapting our tools for the smooth muscle within the uterus. So the uterus is also a muscular organ, but we understand very little about what causes the uterus to contract during labor, for example. And then we've also been moving into the third dimension by both engineering muscle tissue. So here you can see a highly contractile 3D muscle tissue that we've made in the lab. And also partnering with Georgia Quadrato in the stem cell department to develop microfluidic devices for culturing and imaging brain organoids. Okay, so with that, I'm going to pass it on to Shannon.
Thanks so much, Megan. So yeah, as Megan mentioned, our next speaker is Dr. Shannon Mumenthaler. She's Assistant Professor of Medicine and Biomedical Engineering. And her talk today is entitled Organ on a Chip Platform for Study Colorectal Cancer. Please welcome Dr. Mumenthaler.
Can you hear me?
Yes.
Okay, great. It just said it had an unmute button that I had to push. So I just want to thank the Mesh Academy for the opportunity to present here on our work using an organ on a chip platform to study colorectal cancer. And also thank you, Megan, for that fantastic introduction on organs on a chip. So I won't have to go into so much detail in our talk. But first I wanted to just start off with the motivation for this work. So here are actually two colorectal cancer patient tumors where the molecular alterations are very similar, and these patients were actually treated identically. However, you can see that the tumors look vastly different. The one on the right really has a higher percentage of stroma compared to the one on the left. And so when we think about cancer, it really is this highly complex adaptive system that relies on more than just the underlying genetics to fuel the disease. And so in order for us to really be able to study the contributions of the tumor microenvironment, in particular the stromal aspects on disease progression as well as therapeutic response, we really need improved biomimetic model systems to do that. And so as Megan described kind of the advantages of the organ on a chip platform, we really see it as kind of this bridge between some of the existing in vitro models as well as the in vivo animal model. And in particular, some of the advantages around studying colorectal cancer with organs on a chip is around kind of the unique aspects of the disease pathology. So in particular, colorectal cancer, as it's progressing and tumor cells are proliferating, they're actually proliferating into the lumen of the colon. And then once it gets to later stages of disease, these tumor cells then leave the primary site and intravasate into the vascular system and then end up spreading to distant sites such as the liver. And so having a platform where we can really resemble the in vivo tissue architecture is really important. In addition, there's some unique mechanical forces that are found within the colon. The shear stress from fluid flow in the lumen, as well as the cyclic strain that you get from peristalsis motion, are important to recapitulate. As well as the stromal contribution. There's a host of stromal cells that are present at the primary site that are influencing tumor progression. And so having a chip model that is tunable to be able to look at these contributions of the various microenvironmental components is really important. So we're using a chip platform in collaboration with Emulet, which is a biotech company out in Boston. And so their chip device is actually, it's a PDMS chip that has two different channels. So you have your epithelial channel where you can seed your intestine cells on top of an ECM coating, as well as introduce other stromal cell types and your cancer cells. Here in the bottom channel, it's your vascular channel. So we can seed our endothelial cells and they form a tube. Now you have fluid flow in both channels, and then what's also unique about this system are these vacuum channels on the side. And so these vacuum channels can introduce cyclic stretching, which mimics peristalsis. And so here's a video really going Z-stack from the bottom. So here we're looking at the endothelial cells. Now we're getting into the porous membrane up to the normal intestine cell types. And then in green here are our tumor cells forming these clusters. And so as you can see kind of from this video, we're able to recapitulate the 3D architecture of the intestine, and you have the tumor cells that are actually growing into that lumen. And so then we wanted to actually really see if our colorectal cancer on a chip could mimic important biology of colorectal cancer. And so then we did some mass spec-based metabolomics to look at metabolites that might be different between a normal intestine chip and now our colorectal cancer chip. And the advantages of these organ-on-a-chip platforms is the microfluidics capabilities and the fact that we can interrogate media that flows into the inlet and or outlet. And so when we look at the metabolites that are differential between our colon cancer chip and our intestine chip, they actually mapped to colorectal cancer when we used ingenuity pathway analysis. And so this was of high significance in comparison. If you just took 50 random differential metabolites, you did not see this mapping. So this gave us confidence in our colorectal cancer on a chip. And then we wanted to move forward and say, could we start to understand some really important biology surrounding metastasis? And in particular, having a model system where we could look at early stages of metastasis, this intravasation, where cells go from the epithelial site into the vascular channel. And because we can mimic the tissue-tissue interface here, we thought that this was a great model to do so. So here what we're doing is using on-chip imaging. This is actually, if you imagine taking a cross section of the chip and then looking down the length of the chip, here we have our endothelial cells in red, and then forming the tube, you have fluid flow in the empty space here. The white dashed line is actually mimicking the membrane, and then above the membrane is our normal epithelium, which is unlabeled in this case, and then our tumor cells in green. And what you can see is over time, you actually get individual tumor cells that invade the ECM through the porous membrane into the vascular channel and either adhere to the endothelial cells or actually flow out into the reservoir. We can then use quantitative image analysis to actually determine the number of cells that are still in the epithelial channel versus the vascular channel over time and calculate an invasion ratio. And so then when we apply this assay to tumor cell lines that are known to kind of span the aggressive nature of colon cancer, we're able to see that we can differentiate between more or less invasive tumor cells using this model. In addition, we have some ongoing studies in collaboration with Keyue where we're actually collecting the cells that are still remaining in the epithelial channel versus those that have intravasated into the vascular channel to look at the gene profile differences there. And then also coupling that with a liver chip model so that we can study, cells that have invaded into the vascular channel, do they have an increased propensity to actually seed a distant metastatic site such as the liver? In addition, we can collect the cells, as I mentioned, that end up in the outflow and count the number of cells that are there. And we can see here that the more aggressive HCT116s were able to detect tumor cells that have kind of appear to be more like circulating tumor cells and end up in the reservoir compared to HT29s where we don't see any cells. What's also interesting is we can take those cells and then seed them onto further 96 well plates and show that these cells are actually viable and they will continue to proliferate over time. And then when we look at the EMT markers, we actually see that the circulating invaded cells have a higher expression of vimentin and lower E-cadherin compared to HCT116 cells that are on monolayer, really suggesting kind of that these cells are undergoing the epithelial to mesenchymal transition. And in addition, again, one of the big advantages of the organ-on-a-chip platform is the tunability. And so the fact that we can modulate various aspects of the microenvironment, and in particular for this organ-on-a-chip platform over even other organ-on-a-chip platforms, is the ability to mimic peristalsis, so the cyclic stretching motion that's occurring. And so when we actually look at tumor cell invasion, in the presence of peristalsis, we see increased invasion compared to static conditions. And then we're also looking at the stromal cell component. And so cancer-associated fibroblasts are the most abundant stromal cell within the tumor microenvironment and have been shown to play a role in a number of different hallmarks of cancer, in particular the activation of invasion and metastasis. However, really there's been some really important papers that have come out recently to show that maybe not all CAFs are bad. Maybe not all of them are contributing to this invasion or metastatic process. And so we collect patient samples from the Norris Cancer Center, and we're able to isolate cancer-associated fibroblasts, as well as for some patients, normal fibroblasts from adjacent tissue. And we wanted to look at really the functional implications of these fibroblasts across patients. And so then we took conditioned media from these fibroblasts, cancer-associated fibroblasts, and what we were able to show is that from some of the patients where we isolated cancer-associated fibroblasts, they were able to promote tumor cell invasion. Some didn't change invasion at all, and others actually reduced invasion. And I just want to call attention to this sample here, where we actually had the matched normal fibroblast and cancer-associated fibroblast from the same patient. And you can see that there's distinct functional differences between those in terms of promoting invasion. In addition, we've been optimizing how to actually seed the cancer-associated fibroblasts onto the chip so that you can really get the physical interaction between the tumor and stromal cells. And what we're able to show is that you get a similar phenotype when you actually have the physical interactions in terms of invasion, but we also see some morphological changes that occur in how the tumor cells actually cluster and interact with the fibroblasts. So we're looking forward to kind of furthering those investigations to understand how that might, those interactions may change progression or may also alter tumor response. And in addition, so we wanted to kind of take it the next step and say, okay, what's the difference between the fibroblasts, the cancer-associated fibroblasts that are actually promoting invasion versus reducing invasion? And when we looked at alpha-synuclein actin, which is a canonical marker of fibroblasts, we saw that those with high alpha-synuclein actin expression seem to have the increased tumor cell invasion ratio compared to those with low expression. And there's been literature really showing that colorectal cancer patients with high alpha-synuclein actin tumors actually have worse survival. So we're really kind of following up on this finding to really understand if we target alpha-synuclein actin, how do we change the early metastatic spread of these tumor cells? And then I just wanted to kind of follow up with extending this work now with our patient-derived organoid biobank that we have. And organoids are also have really been an important model system in the cancer space, but really being able to couple now the organoids with the organ-on-a-chip platform, we can recapitulate the molecular and phenotypic heterogeneity of patient tissues. And here's just showing an example of taking our normal and tumor organoids and seeding them onto the organ-on-a-chip platform. And you can see that under flow and under cyclic stretching, you're able to recapitulate the 3D architecture of the intestine. You're also able to mimic an intact barrier function. And then we're able to seed these tumor cells on and show that they can actually proliferate in this system. And so in summary, I just want to close with the fact that organs on a chip are really a tunable model system to study colorectal cancer progression, or really any cancer in this case, and the fact that you can tune and alter the microenvironment and really see how do these different microenvironmental factors change the disease progression is really an important step in the biology field. And so with that, I really want to thank specifically a very talented graduate student who's done a lot of the work, and then this has been supported by many others in my lab and collaborators within the Ellison Institute, and as well as others at USC and Emulet, also my funding sources, and just also want to put out there that I'm hiring a postdoc as well for this work. So I will stop sharing at this point and let Keyue go.
All right, thank you so much, Dr. Mumenthaler. So for our final speaker, we have Keyue Shen. So Dr. Shen is Assistant Professor, Department of Biomedical Engineering. He's also a member of the Norris Comprehensive Cancer Center. And his talk is entitled Engineering Cell Microenvironments for Cancer and Stem Cells. So please welcome Dr. Shen.
Thank you, Melissa, for the introduction, and I thank everybody for being here to my talk. And I would like to introduce some of the organ-on-chip models and tools we have developed, and also some other additional analytical tools through imaging in this talk. And before I start my talk, I would like to very briefly thank the people that have actually worked on the projects, my graduate students and also the undergraduate students, and also my collaborators at USC and also in City of Hope and Cedars-Sinai, and also my funding sources. So my lab is particularly interested in two types of microenvironments. We are creating platform technologies to analyze those. This include the tumor microenvironment, which is known to actually drive heterogeneous cancer cell behaviors even within the same tumors and the same tumor type. And also in the bone, we are also interested in the bone marrow microenvironment, the hematopoietic stem cells. We are trying to mimic the bone marrow microenvironment in vitro and trying to expand the hematopoietic stem cells for bone marrow transplantation to solve one of the biggest challenges in the regenerative medicine. In brief, as my colleague, Dr. Megan McCain has introduced, we are basically working on the developing models between the in vivo model and in vitro model that actually recapitulate some of the key features of the in vivo system in terms of the organ, organ organization, tissue organization, some of the complexities in the physical parameters in those cell microenvironments. At the same time, we can maintain some of the main features in the in vitro cultures for their simplicity and well-controlled nature. So my lab is focusing on this cancer and the stem cell area, and then in particular on the tumor hypoxia and the tumor stromal interaction for the mitochondrial heterogeneity. We are also looking at the metabolic features of the stem cells in order for promoting the stem cell expansion in vivo by mimicking the stromal cells in the in vitro culture. So first, hypoxia. Hypoxia is basically a very important feature of the advanced tumors that drives many malignant behaviors in the cancer cells for their drug resistance and also resistance to the immunotherapeutics. So in our lab, we had developed a platform, basically it's a hypoxia microdevice that eliminates some of the very complicated microfluidic control in the common kind of hypoxia device that's developed in the bioengineering groups. So we basically utilize the cell's native ability to metabolize and then generate an oxygen gradient because of the diffusion reaction limited process. So we sandwich a layer of cancer cell between two oxygen barriers, and oxygen can only come from the periphery as they are consumed by the outer layer of the cells. The level of the oxygen in the middle of the device become very low. So there's a natural oxygen gradient and also metabolic gradient in this device. So in this device, we basically can mimic through the computer simulation what the oxygen gradient will look like. And then we have seen that this oxygen level can be reached very quickly within like the first hour of the culture. We can also integrate optical sensors to measure the oxygen level within the device in real time. We can further integrate this micropattern and the micro device with the laser capture microdissection to take cells from different regions of the cell culture to analyze the cells in the different regions of hypoxic field to see their phenotypes and compare against each other. We can also further investigate the drug responses in the tumor microenvironment, how the hypoxia and the drugs can interact with each other. And then some of the drugs, for example, tirapazamine is a hypoxia activated drug, can specifically kill a certain region of cancer cell within the hypoxic field. So this platform can be further used for other types of drug studies in the hypoxia environment. And we have further upgraded the system to incorporate three-dimensional nature of the tumor microenvironment and investigate the interaction of the immunotherapeutics in the tumor microenvironment. Specifically, in this case, we are collaborating with Dr. Ping Wang's group in chemical engineering to look at the CAR T cells, how they get into the solid tumor microenvironment and kill the cancer cell. And then we basically, instead of using a two-dimensional cell layer, we embed the cancer cells in the extracellular matrix and then introduce a microfluidic channel to allow the CAR T cells to infiltrate from the periphery. And we have shown a very interesting cell killing pattern in the hypoxic device, which is very related to the oxygen level within the device and also the physical locations of the cancer cell and also the CAR T cell. We have shown the CAR T cell mediated cell killing is not only limited to the region where the CAR T cell infiltrate in the tumor environment, but also kill the cells remotely through additional mechanisms, mainly through the metabolic interaction between the CAR T cell and the cancer cell. And in that end, we have been investigating on how we can manipulate this metabolic environment in the solid tumors to further promote the cancer cell killing. We are also investigating the identity of those CAR T cells that's actually infiltrating the tumor environment to investigate how we can improve the engineering of the CAR T cell therapeutics to enhance their infiltration or enhance their killing in the solid tumors. And we are further integrating the vascular component in the solid tumor model, integrated with the hypoxic tumors by including large vasculature or microvasculature in the system. We are also further developing microfluidic platforms that can integrate multiple microfluidic channels so that we can actually investigate the therapeutics in a highly controlled way to avoid a lot of systemic errors between different devices. And then we are also upscaling the culture platform so that we can investigate many different microenvironmental factors and the hypoxic degrees in the devices in a high throughput manner. So the other area we are focusing on is looking at the so-called mitochondrial regulation of a cancer metastasis. As a cancer metastasis is one of the major causes, actually the major cause of cancer death in the patients. Metastatic cascade starts with the cancer cell initiating some invasive behaviors in the primary tumor, getting into the blood vessel and then transport to the distant organs to see there to become metastatic tumors. So it's very important to understand what's actually going on in the initial stages of the cancer invasion and the metastatic potential. So people have found in the study that the mitochondria in the cancer cell actually play a very big role in the metastasis. And then people have shown that the circulating tumor cells have a much higher mitochondrial biogenesis or mitochondrial activity than the primary tumor cell. And interestingly, there's increased mitochondrial activity in the cancer cell in the primary tumor, but people don't understand why there's such a regulation of mitochondrial behaviors in those peripheral cells. So in our lab, we developed a micropatterned tumor stromal assay through a stencil-based cell patterning technology, so we can actually lay down the cancer cell and the stromal cell in a very spatially confined pattern. So we can investigate what's actually going on at the interface between the cancer and the stromal cells, and also investigate the remote interaction between the cancer and stromal cell. So with this type of a platform, we have used the RNA sequencing to look at the gene expression patterns within this micropattern culture. We indeed found a signature that's related very much to the mitochondrial activity and the metabolic activities within these tumor microenvironment, and seeing an enhanced mitochondrial activity on the periphery that's particularly related to the electron transport and proton transport. That's pretty much related to the mitochondrial membrane potential in the cancer cell. And we indeed found that the mitochondrial membrane potential is upregulated on the periphery. This has significant meaning in the cancer metastasis. As we investigate in the collaboration with Dr. Ming Yu's group, we found that the cancer cell with a high mitochondrial membrane potential actually can generate higher metastatic burden in animals. So we are investigating basically what's actually regulating the spatial kind of differential behaviors of mitochondrial membrane potential. In one specific direction we're investigating is the biomechanical regulation of mitochondrial membrane potential. We basically micropattern the cancer and the stromal cells or cancer cells alone in different kind of micropatterning platforms. And then we found that the mechanical cues particularly play a big role in regulating mitochondrial membrane potential. As you can see from here, this actually very good correlation between the mitochondrial membrane potential and a nuclear mechanical regulator, mechanical transponder, the YAP-TAZ. They are very strongly correlated with each other in the culture. And then we also further found that the YAP-TAZ actually negatively regulates the mitochondrial membrane potential by an overexpression model. So now we have developed and fabricated a mechanical stretching device. We can actually directly investigate how the mechanical cues can directly influence the mitochondrial membrane potential in these cancer cells. And the other very important direction, as I mentioned earlier in my lab, is to investigate the hematopoietic stem cells and its bone marrow niche. Basically, our long-term goal is to develop in vitro platforms so that we can expand HSCs for the bone marrow transplantation. So one of the big challenges in this field is basically how we can actually monitor and actually have a quick feedback on the stem cell features of the hematopoietic stem cells, which is still very lacking in the field. So one of the key features we focus on is the stem cell metabolism, which has been shown by some previous studies through the mass spectrometry that the HSCs has a very distinct metabolic feature compared to the progenitor population. That in part is coming from the highly hypoxic niche they are coming from in the bone marrow. So the HSCs has a very high glycolysis and a very low or minimal oxidative phosphorylation in the mitochondria, which is referred to as a metabolic stemness by some publications from Dr. Suda's lab. And then we are basically focusing on this particular metabolic stemness feature and then using a fluorescence lifetime imaging microscopy technique that has been that was actually available through the Translational Imaging Center led by Dr. Scott Fraser's lab. We are collaborating with them to look at the HSC metabolism as compared to the more differentiated populations. We have shown that actually using some non-invasive optical biomarkers in combination through principal component analysis, we can distinguish HSCs from the more differentiated populations. And then more importantly, we have also developed a machine learning model to utilize those non-invasive markers without the assistance of other markers. We can actually identify the HSCs directly from the progenitor populations in a very closely related multipotent progenitor population with very high sensitivity and specificity. So with this type of technology, we have further developed this non-invasive biomarkers into a consolidated marker to define the metabolic stemness quantitatively. So we basically combine those parameters and use a PCA analysis and then find the principal component that's actually underlying the glycolysis and oxidative phosphorylation. And then we can conveniently define the HSC stemness based on this so-called MOB score. MOB stands for the metabolic optical biomarkers we have previously developed. And then we can actually monitor also accurately how the differentiation is actually going in the in vitro culture. And then we have been utilizing a micropatterning platform to pattern single HSCs and monitor in the long term how the HSC would divide on the single cell level into daughter cells and how the stemness are maintained in these daughter cells. We can monitor basically the symmetric division, which will give rise to an increased population size of the stem cell, or the asymmetric division that will give maintain the level of the population in the stem cell, or the symmetric differentiation in the population. We have found like very interesting behaviors of the stem cell in the division. We are utilizing also metabolic interventions to actually try to enhance the symmetric division of the stem cell and maintain the stemness features in the in vitro culture as a way to expand the HSC populations. On the other hand, we are also very interested in mimicking the stromal environment in the bone marrow in order to expand the niche size, basically that dictates the number of the stem cell. And by mimicking those niche conditions or parameters, we can actually significantly increase the stem cell population in vitro. So what the very important thing is what's important in the bone marrow niche. We are focusing on one particular type of feature in the bone marrow niche, which is the membrane factors on the stromal cell, which is critical for the HSC maintenance. We are mimicking them on the lipid membrane to mimic their molecular features on the stromal cells. And we have found that some of the membrane-bound factors are very uniquely promoting a polarized morphology of HSC, which we suspect actually has a very important role in this stem cell migration or in stem cell homing to the bone marrow niche. And at the same time, these membrane-bound factors are also critical for promoting the stem cell maintenance. So in this stem cell work, we are trying to further develop a couple of technical platforms. For example, using a cell microarray platform to isolate the stem cells based on the metabolic features non-invasively. And then we are also developing techniques to investigate how the membrane-bound factors are actually influencing the stem cell division, and then to further promote the stem cell populations in the in vitro culture by looking at these metabolic features in the daughter cells. And we are also developing microfluidic platforms to look at how the stem cell morphology is in regulating the stem cell migration and then homing to the niche environment to promote the transplantation efficiency in the bone marrow transplantation. And then we're also developing a materials platform to fabricate so-called artificial niche stromal cell that contains both the membrane-bound factor and the soluble growth factor to actually promote the stem cell growth. With that, so I would like to conclude my talk and then turn it to the meeting organizer.
All right, thank you so much, Dr. Shen. So we'd like to go ahead and open up the floor for questions. As I mentioned before, we really encourage you to raise your hand and I can call on you. You can ask your question directly to our speakers. If not, you can go ahead and put your questions in the Q&A. I see we have a few in the Q&A. I guess I can start with those to begin with. Let's see. So first off, we have a question. This is Kwanjin Kim, and this is for Dr. McCain. Any attempts to study the interaction between vasculature and your model?
Thank you for the question. So we haven't done that yet. We've been looking at interactions between cardiac myocytes and fibroblasts, and we've been looking at interactions between muscle cells and neurons. And so we're also interested in looking at interactions between both cardiac and skeletal muscle with endothelial cells and vasculature. We do have one project that's just starting to look into that, so stay tuned for that. But maybe the other speakers also want to comment on that as well.
Any thoughts from anyone else? Any of the speakers?
I mean, we can comment a little on, I think that's one of the great aspects of these organ-on-a-chip platforms is the tunability, right? And so to be able to add in various cell types and really study the crosstalk that's occurring. So yeah, we're definitely in our colon cancer model looking at different stromal cells as well.
Yeah, great.
Yeah, so in my lab, we are also kind of like integrating the vasculature. Although it's still in the early stage, we are kind of putting the vasculature as a delivery kind of channel for these cells. But people have also shown the vasculature itself can have a biological kind of interactions with the cancer cells, in those cancer co-culture models. So that's definitely another very important factor.
Yeah, I think I didn't touch on it, but I had the data in there. We've looked at kind of the chip model without the endothelial cells, and that also influences the tumor cell invasion. So there's clearly kind of secreted factors as well as potentially other cell interactions that are occurring that are influencing that invasion process. So I think having the vasculature in there is going to be important for the cancer studies as well.
So I have a second question here for Dr. McCain. This is from Ashutosh Argawal. So the question is, do you foresee any value in coupling your work with neuronal tissues with uterine smooth muscles as well? Perhaps to trigger contractions? Maybe I'm way off.
Thank you for the question. It's actually a provocative question that I'm going to think some more about. So with the uterus, generally we think of hormones as triggering contractions like oxytocin or progesterone suppressing contraction at different stages of menstruation as well as labor. So what we're focusing on in my lab is also looking at how different mechanical stimuli like stretch can stimulate contraction. But I think looking at also neural input could be interesting. And I'm not sure there's a whole lot known. The uterus, really, we know very little about it. It's pretty shocking. But there's been some interesting work, for example, in the heart. The heart, you can take the heart out of the body and it will continue to contract. So neuronal input, the way that skeletal muscle do to really drive the contraction. But there's been a lot of interesting studies in recent years showing that neurons can also affect or kind of modulate the contractility of cardiac tissue. So yeah, I think it's a really interesting question and something that I would be very interested to look at in our system.
All right, great. So again, anyone have any questions? Don't be shy. This is your chance to speak to the speakers directly.
Can I ask a question?
Yeah, yeah, go ahead.
So Keyue, I was wondering if you've done any of the phlegm imaging on your micropatterns when you do the tumor stromal patterning. Have you actually looked at kind of the metabolic interactions there, and especially on the edges where you get the physical interaction?
Yeah, so we have actually looked at use the phlegm to look at cancer cells as well in this problem. That's a very good question because it's natural. You have tool to look at a metabolism in the stem cell, but that must be true for the cancer cells too. We have found that actually in one of our early publications, we have shown with just the optical redox ratio, it's a very traditional marker to show that the cancer cell on the periphery near the stromal cell actually has high mitochondrial metabolism than the cell in the center of the micropattern. Some other additional parameters are a little bit harder to interpret. We're still investigating what's the mechanism behind them. Some features, those markers are actually very, I would say, be cautious about interpreting a lot of the optical biomarkers. You have to actually do it in the cell type specific way and then use some good control conditions to investigate that. So we have seen interesting features kind of associated with the cancer cells like in these different regions, but we don't exactly know what's the meaning of that. The only thing we can interpret and with high confidence is optical redox ratio so far.
We should talk more because we're doing some phlegm imaging, especially with our organoids, and briefly started doing some in the chip model as well. So I'd be curious to kind of talk about some of the advantages as well as some of the challenges as you start to add on the complexity in these model systems.
Mm-hmm.
All right. Let's see, I have a question here from Steve K. So this is for Shannon. How much in-house experience do we have with liver 3D culture, given the interest in HCC and other liver diseases at USC?
Hi, Steve. Yeah. So what's nice about the chip platform that we're using is Emulet has also made a normal liver chip model, and so it's using the same PDMS base, but it's just different cell types. And one of the big challenges, especially with developing kind of a normal liver, is the sourcing of cells. So they've done a really great job of really having high quality cells to be able to seed the liver chips. And you can really do anywhere from kind of a co-culture where it's just your hepatocytes and your liver sinusoidal endothelial cells up to kind of a quad culture where you add in the Kupfer cells and stellate cells. And so we've been working with those models, and in our case, kind of using it as a model to study the tumor cells that have been travasated into the vascular channel. So those kind of highly invasive cells, are they able to eventually seed onto the liver and colonize that distant site? But it could be adapted very easily to kind of HCC as well as kind of other disease models for the liver.
Great. All right. Anyone else have any questions? We still have time, so please ask your question if you have any. I guess you guys had really great presentations and explained everything well. So, oh wait, we have another one. Let's see here. All right, this is from Unmesh Havdar. Sorry if I'm butchering your name. So this is for Shannon. Oh, okay. This is Unmesh from the Stem Cell Center. We just started our lab and have interest in epigenetics of colorectal cancer. Have you worked with mouse organoids in your model?
We have not, but it's something that actually we were just starting to think about. Everything we've been doing has really been at the human scale in terms of our organoids, but I would be very interested actually in speaking with you. I'm aware of your work as well, and would love to kind of follow up, because I think there's some unique work that you can do, especially with the animal models that you may have, and being able to do some different engineering of those organoids to put onto the chip model and study kind of epigenetic interactions and stuff there. So we have not done any mouse organoids, but we would be interested in following up.
Great. All right. Any other questions? Would anyone like to ask a question directly to our speakers? We have a very shy audience today. All right. Well, I guess if there isn't any additional questions, I guess we can wrap it up. So I just wanted to thank all our presenters today, Dr. McCain, Dr. Mumenthaler, and Dr. Shen. We really appreciate you taking the time and speaking with us and telling us about your great research. I'd also like to thank all of our attendees for joining us today. We appreciate, again, you taking the time and joining us. And then we really hope that you can join us for our next seminar, which will be November 5th. So with that, thanks everyone, and we look forward to seeing you next time.